Good morning, everyone, and welcome back to the second session of OmicsLogic Data Science. How is everybody doing today? So before we start today's session, I wanted to do a quick review of what was discussed yesterday and see what the progress have been made together as a group. So let's go and see the progress together. So this is the organization page again. I'll put that down in the chat. And as we see, since yesterday, there has been quite a bit of progress right here. So you see the graph is going up. And now let's look at the participants who have been doing some of these different things. So I see that Sangeeta has been running some of the server platforms. So she has 500 points now. Good work, Sangeeta. And same as Nagaraju, he finished the Introduction to Bioinformatics course, which was one of the assignments from yesterday. So he has also received 500 points. Here you see Renuka. For the rest of you, you all have not started, and that is okay. You can still take your time and do it. But as we will start talking more about the other courses, if you're not finishing with Introduction to Bioinformatics, then it will become even more harder to keep up as we go forward. So let's look at some of the individual profiles. So here, Renuka, good work with your profile. So right now your profile is completed. You have a full name and a profile photo. And you can see that there is 450 points that she has earned by completing a pipeline on the server platform. So this is where with individual profiles as well, we can track each and everyone's progress. So if you are not doing any of the assignments, we will be able to see it very clearly on here. Same here for Nagaraju. You see that I left a comment on his profile as well. So you can also comment, you can favorite someone's profile, you can actually interact with each other on this platform. So start doing that, start exploring the courses, start talking and discussing about different things on the forum. And we would want to see more and more interaction from your end. Same here for Sangeeta. Good work, Sangeeta. But again, Introduction to Bioinformatics. That was the assignment. So I see that not a lot of you have done. Here I see Surya has completed his profile, but has not started with any of the courses. And how do I also know that your profile is complete or not completed? I can actually look here and I can understand. You see here, Renuka's name is appearing in full. And if your name does not appear in full, it would be like, say, for example, just an username. And you do not want that. The reason you do not want it is because, see, Nagaraju has completed. So once you finish a course, you also get individual certificates for these courses. So you can collect as many as, say, if you do 8 to 9 courses, you can collect those many certificates, right? So if you go, you will be able to see that under each course, it is a completed, it's been highlighted in green. And if you are doing a course, you will be able to see the progress, like how much progress you have made yourself. So currently, say for example, he did not start with transcriptomics 1. So the course result is 0%. So it will be just, so you will be able to track your progress throughout this entire journey. So let us look at the certificate that comes up. And the reason that we always tell everybody to complete their profiles is because this certificate, this is not the program certificate, but again, the individual course certificate is generated for each of these courses you will take. So because Nagaraju had already done Introduction to Bioinformatics, you can see that his certificate has been generated and he can actually download this certificate as well and share it in LinkedIn or add it to his CV. So his name is the same as what he has mentioned here. So please keep updating your own name, and similar way you will also be able to find all of your certificates here once you start completing the courses. So by next time, I hope that everybody would be completing their profiles and getting started with Introduction to Bioinformatics. And now I would like to pass on the session to Dr. Harpreet. Dr. Harpreet, over to you.
Thank you, Vipsa. So, hello everyone. So, in continuation of to the this program, in today's lecture, we will be focused on some of the major challenges that are presented by the large data or big data and requiring special attention to the exploratory data analysis and domain expertise to help separate technical noise from the biological signal in our data. We will also explore how major research principles of reproducibility require us to develop a deep understanding of the methods we use and incorporate a clear record of setting, data transformation steps, and outputs. So before heading into today's session, let me review what we have learned in our last session. So in our last session, we started with the history of the genomics data. Then we learned about various types of the genomic data. Next, we understand of the central dogma of the biology and just get an overview of transcription. Then we provided you with an overview of the splicing. Next, we learned about the different methods that are used for quantification of the mRNA. Then we learned about the NGS RNA-seq technology. Subsequently, we learned about various steps that are required for RNA-seq analysis. Then we learned about different algorithms that are required to perform different NGS RNA-seq steps. Next, we demonstrated to you how we can run a pipeline on T-Bioinfo platform to produce a gene expression table from the raw reads of NGS data. Then we understand about the various aspects of a table, what it represents, and where the samples and the genes are. Next, we look into the genomics data, specifically somatic mutations. Then we understand about various types of the mutations. It could be a point mutations, then based on the impact they made on a genome, like missense, nonsense, and frameshift mutation. At last, we provided you with an overview of the metagenomics. So at the end of last session, we have asked you to complete some of the assignments, and we have looked that you yet not started. So if any one of you able to complete at least, I think only we have complete your profile was our first question that we want to ask, that you should complete at least before the next session. And then introduction to the bioinformatics and introduction to the genomics. So if most of you have yet not started, please start completing these courses. Otherwise, it would be difficult for you to cope up with all these sessions. And of course, explore the T-Bioinfo platform. I hope today every one of you able to just log into your T-Bioinfo account. So just let me know how many of you already able to log into T-Bioinfo platform. So, okay, so I think
Dr. Harpreet, there are a lot of people. Okay.
Anirudh, I think Anirudh have he's saying he had already finished, but it's not reflecting. Might be you have not completed the course. You have to click a button that was saying finish the course. So you might have forget to click that. Otherwise, it would be shown on your profile.
For the pipelines, demo pipelines that we tried in class yesterday would not always be reflected on your profile, just saying, but there will be assignments going forward in transcriptomics 1 and 2, where you will be asked to do some assignments and upload data. And all the course progress would be reflected on your profile. And the pipeline URL is the same URL you will be finding on server tbio. Whenever you're starting a pipeline, there will be the URL will be changing. So that is the URL you can always share with us, so that even if you have some problem, we can look into it. So it will help us troubleshoot that. Okay, is that clear for everybody?
I think these are demo pipelines. That's why it doesn't, show it on activity. It is just, the point is that, so the demo pipelines might not be shown on in the activity. So, okay. So now let me ask a few questions. So let's see how much you learned in the last session. So first question is, for which file format represent the raw read? FASTA, B, FASTQ, C, text format, D, GTF format. So just post your answer in the chat box. Sai Kabiri saying FASTQ, FASTQ. Okay. Yeah. So I think, yeah, most of excellent, you all are correct. It is the B, that is FASTQ. So it contains raw reads as well as the quality score for each of the nucleotide in the that is present in a sequence. So the next question, which file format represents sequence of the genes or the proteins? A, FASTA, B, FASTQ, C, next text format, D, GTF. So which file format represent the sequence of a gene? Gene or a protein? FASTA, Ankita Singh. Okay, so excellent. Yes, it is the FASTA format which is used to present any sequence of a gene or a protein. So which file format indicate the position of the exons and the introns and used for the annotation? It's FASTA, A, B, FASTQ, C, text, D is GTF. Which file indicate the position of the exons and introns and used for the annotation? A, FASTA, B, FASTQ, C, text, and D, GTF. Okay, so Anirudh saying GTF. Srinivas saying FASTA. Anyone else? Bharat saying FASTA. So Anirudh is correct. It is the GTF file that is used for the annotation, and it represents the position of each element. It could be exon, introns, or any gene So answer is actually D, that is GTF format, and it is this GTF file is used for the annotation. So the next question, which of following are two algorithm that can be both used before mapping? 1. A, PCR clean and Trimomatic. B, clean primers or isolate. C, eco and the agile. D, Botai2 and PCR clean. So which of following A? So, Preeti, Shreyas, Keri, Yukta saying E. Yeah, all of you are correct. It is PCR clean and Trimomatic. As we have already told you that PCR clean removes the PCR duplicates and Trimomatic removes the adapters from the sequences. These are pre-processing steps. So the next question. So looking just at this pipeline, so what output you will get? A, PCA plot. B, differentially expressed genes. C, gene expression table. D, genome visualization. So Sresh, okay. Yeah, excellent. So with this, we can generate a gene expression table. So now let's begin with today's session. So what were the properties that makes our data as a big data? Now let's understand what are those properties. So there are the 5 ways that may describe a big data. The first is volume. So the amount of data that is used. For example, we know that RNA-seq yield an information about thousands of the genes or millions of the single nucleotide mutations variants. Then the next is variety. The data is unstructured and can be assembled in many different ways. Often at times, the same phenomena can be described in a different ways depending on the type of the sequencer, preparation techniques, or handling in a lab setting, and of course, depending on the person who is designing that experiment. Then the value. Third one is value. So here the information we get out of any data is dependent on many aspects, including data noise technique, then error rates, and then completeness of information a single sample that contains. Then fourth one is veracity. That is, some samples can have low coverage and others have high coverage. So as I have already told you regarding that, but not the full sequences. Others might be generated by old machine that had high error rate. So even so, all including all the data together can complete this whole picture regarding a particular sample. Fifth one and most important, that is velocity. That is one of most important aspect, as we can see it is changes in the data types and the new information that is generated by it. So for example, with time, Sanger sequencing is replaced by the shotgun sequencing, or that is an NGS. And eventually, nowadays, a single cell sequencing comes into the play. So we are always learning new things and have to adapt to this idea that new samples expand our understanding before even we had any chance to study everything from the data that we already have. So these are the major factors that contribute to a major properties that eventually makes a data into a as a big data. So as most of all of you are from the biology background, that is biochemistry. So what would be your a biologist perspective whenever he start proceeding? So a biologist is trained in the basic biological processes. So then they specialize in certain area where they gain both knowledge and experience from the experimental data. So many of the examples we learned about, we learn and we get the information that accumulated from carefully designed experiment was a certain based on certain assumptions. So the generated data from these experiments confirm some of these assumptions, and those were recorded as proven hypotheses. So eventually all the biologists, what they do, they submit their experimental data in different public repositories. So as a result, so here lot of data from experiment is available, and this data is continuously growing almost in exponential manner. So since the discovery of the human genome project, so this data is continuously start growing at exponential rate as one of the major repository, that is NCBI. So if we talk about till today, it have more than 80,000 public data set that's spanning hundreds of the tissue types in thousands of the organisms. So the grow as we can see in by the year 2013, there are the data that are available in different repositories about 153 exabytes. So by 2020, it becomes like more than 2000 exabytes. So this is how this data is continuously getting growing. So the growth in this high throughput sequencing of cancer genomics and transcriptomics has produced such a big data problem that precludes many cancer biologists or oncologists from gleaning knowledge from these data regarding the nature of the malignant processes, as well as the relationship between tumor genomic profiles and treatment responses. So that is why it is important to learn about all these data types and how efficiently we can process and analyze such a huge size of data. Then the next is cost concern. That was driven by obviously a costly research. So, but however, there is gradual transition to effective technology that was scaled up and addressing this cost concern regarding the research. So this issue, the use of this data, the next issue is how we can use such a large amount of data. So how do you analyze such a rich data sets? So many measurements yield a single number of each observation. For example, here we can see it could be height, could be in inches or the centimeters, by weight could be in pounds or kilogram, age could be in years. So using such number you can learn something, but the details and the depth of the information still remain limited. So it is also initiative here on this. If we look at this particular chart, so we can see a clear separation in two dimensions. So weight is projected on y-axis while size or the height is projected on the x-axis. So a straight line separate between a male and the female. While on this is like a low throughput data analysis. While in comparison to in case of the high throughput data, that is the omics data that we are talking about, or big data, so it yields thousands of the data points for each sample, or each element. Here we can say like a gene. So it is associated with its expression. So if we talk about today, there are more than 20,000 protein coding genes in the human genome. So to work with such a high throughput data, one must rely on the methods that can handle this complexity. So let me ask what kind of data types that you know. So just I am asking a question that here are two types, that is categorical and quantitative data. So can you give me any example of categorical data? Anyone can give me an example of categorical data? What you mean by a categorical data? Do you understand? Okay, if you doesn't know, let me give you a very so category means there are the class. So categorical data can be a binary, can be nominal, ordinal. So for example, we have a clinical data. So in the clinical data, for example, a sample can be a if I'm talking about the cancerous condition, it can be a cancerous sample, it could be a normal sample. So this, apart from that, a disease condition can be a early stage, can be a late stage. So within even the stage, we can say it could be a not just a binary, it could be a stage 1, stage 2, stage 3, or stage 4. So these kind of data are coming under the categorical data. So then there is the quantitative data types. It could be a discrete or it could be a continuous. So can anybody give me an example of a continuous data? A very simple example. So do you have any example of continuous data? So a simple example could be height of a people. Yeah, that's a great, Renuka. Yes, height of a people in a population. It could be age, height. Then if we talk about high throughput data, it can be gene expression data, right? So those are coming the numerical data that is that can be measured or can be quantitative, quantified. That those kind of data is coming under the quantitative. So like gene expression value, right? Protein expression value. So those are quantitative data. So beside these kind of data, then based on the data dimensionality, then we could have univariate, bivariate, and multivariate. For example, univariate, when we have only one variable per subject or a per sample. So it could be one variable. I'm just taking an example of a single gene. Suppose I'm talking about a single gene. So here, as I have a one gene, I could say it is univariate. Then I could say a bivariate. So bivariate could be I have two factors to compare or two variables. So those when we have two variables, like even in a simple example, we can say for a human, just a person, it could be age or gender, it could be age or weight, that could be the variable. So then it could be bivariate, then it is multivariate. So if you are considering multiple factors, so like for a person, a very simple example could be variable. It could be like you are considering its age, you are considering its weight, it could be considering its height, then you are considering its eye color. So if it's more than that, so then you are considering saying that there are multiple factors or multiple variables. So that kind of data is called multivariate. So then in case of the gene expression, we could have large number of the genes, large number of uh, large number of proteins. So those kind of data, especially in case of big data, we consider them as multivariate data. Linked to different data properties. It could be its type of data, or it could be based on the dimensionality. So then there are various statistical approaches that are used for the analysis of a data. So the purpose of statistical analysis is to learn more about some population of interest. So like it is just impossible that we have access of every individual in a population. So what it actually we does, so we typically take a sample of subset of some individual from the population. So with this sample in hand, we seek either summarize this data using various statistical descriptive approaches. So, or use this data to make any prediction regarding this particular population from which this particular sample of data has been taken. So these traditional methods of statistical analysis were developed to work with the demographic, so astronomy and biological data. So all of these domains have been transformed by the emergence of the big data, where each sample can be described by a thousands of or millions of the data points. So that can be generated with the increased speed due to the automation. So statistical analysis methods, in number of the ways they help us to develop an initiative representation of the data properties, or helping us to find out the relationship between various types of the data that we have in our particular desired samples or subset of the samples. Then each type of data can be visualized, focusing main focus on that is the trend that is important within the selected data. So once data set grows, so multivariate analysis method can help us to discover the trends in within the big data, and they can describe the complex relationship between the data points in each of these samples. So before, let me ask a simple question. Do you know what are different resources from where you can obtain a big data? Can you name me any resource from where you can get a data? So I'm talking about the biological data. So from where you can get the data? So just name me any resource that you know. PDB from NCBI. Madhuri Singh. Okay. Okay. So can you tell me a particular resource at NCBI? So as you are most of you saying NCBI, that's a great, excellent. Of course, NCBI is a major resource. Can you tell me a specific name the specific resource at NCBI from where you can get data? Can anyone name a specific resource, as NCBI is a mega resource, so it have lot of resources. Can anybody name a resource where you can find the data? GenBank, that's excellent. Renuka. Anyone else can name a resource from where you can get data? I think already EMBL. Okay, that's excellent. Neha. Okay, so yeah, there is NCBI's major resource. So the first is, let's go to the, if you want to understand the want to get a data related to the research publications. So let's go over these resources. Just let me just give an overview of some of the resources here. Then we will go over to these resources and we will see what kind of data is available in these resources and how we can access different kind of data. So for the publication, if we want to look into the, and the most important aspect, what kind of data that we are looking in a resource. So depending on the our desired data, so there are different resources. For example, if you're looking at the data related to the publication. So one of the major resource whenever you go for to search any publication, there is a major resource that is PubMed. So then there are different resources at the NCBI, as I have already told you, that where you can find different omics data. For example, the GEO, that is gene expression omnibus. So here you can find various types of omics data, like gene expression data. It could be a methylation data. It could be a mutation data. So different types of omics data is available at the GEO. Then the next major resource at NCBI, that is SRA. So that is sequence read archive. So if you need a raw sequencing data, you will go to this particular resource, that is SRA. Then there is a data resource, that is bioproject. So it contains the information related to a particular project, and then you can find different types of the samples here. So beside, can anybody name me any other resource so where you can find biological omics data apart from these NCBI resources? So do you, any one of you can name a resource where you can find omics data related to any disease or any other thing? So, okay, if you don't know, so there are some of the dedicated repositories, like for the cancer, there is the Cancer Genome Atlas, that is TCGA. Then the GDC data portal, where you will find various kinds of omics data related to 33 cancer subtypes. And apart from that, then it is cosmic repository, where you will find mutation data related to the cancers. So apart from that, then our dedicated repositories on the viruses, that is NCVI virus. So here you can find data related to the virus, different viruses. So now let's go over these resources and look what kind of data we can find in these repositories. So now let's see what kind of data is available and how you can select a particular information and how you can search for your data. So in the PubMed, if you want to extract the publications in the topic that you might be interested. For example, I want to look at a data like liver cancer. So if you go there, then you will find, even based on the if we looking at this left side, we are looking at a word plot. So it will indicate it with the every year how many research publications are there. Like if we look at this, so it's around 1880. It was only just a one publication. So by 20 nearly, this is by 2020. We have more than 10,000 publications related to the liver cancer. So then how you can search and define a specific field? So then you can go to this advanced option. So under the advanced option, so you can define a specific terms, like here I am just selecting liver cancer. So suppose I want to extract the publications that are related with the liver cancer and the biomarkers. So here, from here you can select the even the field. So where you are looking for, like here we can search in the title, then we can search under the title and the abstract. So for example, we just searching for the title and the abstract. So then you can add other terms. So other term might be biomarker. So this is how you can extract the various kind of publication that you want to understand. So then you just search for this. So it will give you the all the publications where liver cancer and the biomarkers terms are present on the title and the abstract. So now here you will find nearly 740 publications. So this is how you can sort out your desired, you can make a desired query based on your research interest. So this is just a first publication. First, when you go for, you want to search for a particular publication, you want to design a query for the publication. So beside, here you can even in the site you can find your data related to like up to last 10 years, the 5 years, then you can also define a range. Then you, if you want to exclude, like you want to exclude reviews or systematic reviews, and then you can select all those different field, the different fields, desired field that you are looking for. Then let's go to the geo, that is gene expression omnibus. So if we go over this geo, first of all, you need to search for a keyword, or you can even provide a geo accession number. So as if you are new to this resource, so obviously you don't know about geo accession number. So you can search here with your only the keyword like liver cancer. So I am making a query related to this. So it will give me like there are the 40, nearly 44,000 nearly 44,000 results it give me. Then I can, here I can select a particular organism. So like I just want to search on Homo sapiens. So I can select them. Then I just want to like, here you can decide whether you are looking for the strain attribute name, whether you are looking for the tissues or the strain, and you can customize it here. So then I'm just looking for, like here I'm just looking for the tissue. So then you can uh, study type here, what kind of data that you are looking for. So we can customize here. It will give us the, it will give us a different options, like whether I'm looking for the gene profiling, expression profiling by array, expression profiling by high throughput sequencing. For example, I'm just looking for expression by high throughput sequencing. So I am uncheck all the other options. So depending on your query or research design, you can select that. And now I can select, if I select on this, so I will come sort this data up to the 109 expression data set. So here the data set that you can find a different data set. So now let's the other term, it will give us an option whether we want to sort based on the number of the sample. So here we can decide our sorting order, like I just want to that it should be like descending order, so high to low number. So I have selected this. So then if we looking at the first data set, here we will find we have the 344 samples for high throughput sequencing. So if you click or select this particular data, so here you will get a detail. So like in organism, so what was their study design? It's a, as it is a super series, so you will understand better the super series or the sub series. Super series when multiple series are combined. So otherwise the independent series. So then you will find what platform they have used. Then but here you will find different samples. So even if you're looking at just a one sample, so you just click on this sample. So then you will find complete detail regarding this particular sample. So this is just I'm looking at a one sample. So obviously when you doing any research, it's not about regarding one sample. So you will go for the complete files. So from here you can go this go to the each individual series. So here you can find a even series matrix data. So apart from that, you will get you can also get the raw data that is usually available in the supplementary file. So you can download these data. So then we, from here we can also go directly to the bio project. So let me just go to this bio project. So once we are here, we just directed to the bio project as it is. So here you will find other details regarding this data. So this is how you can go to these bio projects. So the next is, let me just do one more thing. So if I click on all the samples, here you will also can get directed to the other resource. So let me just click on one series. I just So then there is SRA. So this is the first geo, and the other important information. Let me just, before going to the next, just so apart from the BIOS, other resource, here you can also direct it to the SRA. So I'm just, I'm later going to the SRA, sRNA selector. So we can select the supplementary data. So let me just tell you one more information. That is, in the series data, you will also, in the series, what is in the series data? It is usually the processed data that is processed by a particular research group that have submitted this data. So in series data, you will also get the more details regarding data, like its clinical and demographic factors. So this is the raw files, already processed files that were, you can download this data. So this series matrix will give you the gene expression that was gene expression, or whatever the data that we are getting, that was processed by already original authors. And then this series file is very important because it also contains the demographic and clinical information. Now let's just go to, now let's go to the SRA. So this is the SRA. So you can directly even search. So in the in Geo, we have kind of processed data, but if you want to search for the raw files, especially raw files like that you are interested, like FASTQ files. So this is sequence read archive, so that you can find the read sequencing data at the SRA. So for example, here we can search for a data. So here you will find, like there are different large number of the sample that is available for the liver cancer. So if you're looking, so if you just interested in this particular study, so here for each sample there is RNA-seq data in the FASTQ file. So if you're looking at, just let me just zoom it. So if you're looking here, so you will find actually this is a zip file that is nearly 4 GB. And actual this data is nearly 10.5 GB data. So if, this is for just only for a single sample. So then how you can select all samples together? You can download a, I mean, it's very, again the challenge is to handle this such, first is to handle this data, and of course as this is a very large data, so how you can actually download or store this data. So those are the challenges always come with the big data. So for those, how to store and this data, as you cannot download such a high data, that's gigabytes or terabytes. So there are the cloud storage like our platform, T-Bioinfo platform. You need not to download your data. So you just need to provide a file. So like here, just go to the send. So just collect a file, and then here you can also just search a summary. Then here we can get the URL of these files, or we can get a run table. So just click on a run info. So it will give us a run table for this SRA data. So let me create a file. So I will show you that how we can just, we need not to download all these files. So this is how we can just download SRA data while it is getting downloaded. So again, the challenge is to handle this such a large data. So meanwhile, if you have any question, you can post it in chat. So these are kind of different data repositories from where you get your data for any kind of analysis. Okay, so this is sRNA table that you have obtained. So this you can directly upload this SRA table on our server. So to process any kind of information, when you have that research account, when you want to perform your own analysis, so this is SRA table. It will give you the run ID. Then you will get the what kind of spot, then what kind of the size, what is the size of this data, then so then it will tell you what kind of data it is, whole genome data, then it is parent data. So this kind, then but the platform they have used, that is Illumina. So then apart from that, so then you will also understand what kind of organism or scientific name. So it's taken from the musculus. It's a liver. It belongs to the liver. Then this kind of information is available in the SRA. Then it also let you know what's the gender of that particular organism. So then whether it has tumor or not. Then so all those information is available in the SRA table. So you can process these data on even T-Info platform. So this is till now this is related to all the different repositories. Apart from there, if you're interested in so now let me go to the one more repository, that is NCBI virus. So if you are interested in the research related to the viruses, so then this repository is one of most useful repository. So here you can search via virus, or so let me just search this. So like here we are interested in SARS-CoV-2. So it automatically, okay, so it will take you to this page. Okay, so here you can find various kind of data. So like here you can find nucleotide data, protein sequencing data, reference genome data. So then you can select that particular data. Suppose you just interested in this particular sample. So first look at so if you're interested in this, then you can go to the download and then you can select whether you want to a nucleotide sequencing, protein sequencing, so that you can define here. So as I don't want to download this data, so now let's, apart from that, you will find various kind of options here, like based on a sequence length, you can even already define your range. So then sequence type, so whether you are interested in GenBank sequences or the reference sequences, then you can also select whether you want only the complete sequences or all the sequences or only the partial one. Then other the protein option provide you whether you are interested in some particular protein or like spike protein or any other protein. So then based on even the geographical reason, whether you wanted to take the data for the Africa or different region, Europe, so American, so all those kind of options that you can select. And then it will also provide you the whether you are interested in specific host. So this kind of info, like if we want to check in humans or the bats, so we can this NCBI resource give you complete information, so give you different options to select your desired or your desired data based on your study design. So you can also download DNA nucleotide sequences, protein sequences, and other reference genomes. So this is all about the resources. So if you have any question till now, so you just ask your questions. So you can post your questions. So if you have any question till now. So I would ask you to please stay active. I mean, that's how we will understand if you are getting something, or so keep it interactive. It is not just like I'm just giving you any lecture. So just be interactive if you have any questions so far. So apart from the resources, so then apart from the resources, then as we have already told you, there are a lot of publications in which the data is available. So we can even select, apart from these resources, we go to individual research publication and find out their data. So we call that as the different projects from which we can get data. Omics, specifically omics data. So during this program, we will explore the examples of the cancer research, viral infections, and microbiome composition that have major impact on all aspects of the lives. So these projects will give you an insight into the data set that you can start exploring to develop a research questions about how these conditions are diagnosed and what causes them to appear and how they develop and how to treat different conditions, like could be a cancerous, it could be like acne and a food intolerance. So let's look at some of the projects from where we have obtained the data for our program, this program. So this is a research publication. In the year 2016, there was a research article that was published in the OncoTarget. So where Bradford et al. Have explored whole transcriptomic profiling of patient-derived xenograft model as a tool to identify both tumor and stromal specific biomarkers. So they investigated RNA-seq as a hypothesis-free tool to generate independent tumor and stromal biomarkers, and explored tumor stroma infection by exploiting the human murine compartment specificity of the patient-derived xenograft or the PDX. So in their study, across a pan-cancer cohort of nearly 79 PDX models, they determined that most stroma can be separated into distinct clusters where each correspond to a specific stromal cell type based on the omics profiles of the samples. So in their study, authors have explored various kinds of cancers, including breast and the lung cancer subtypes, using the PDX model. So complete publication is available at this particular URL. So as we are talking about the patient-derived projects, now let's understand what is the PDX model is all about. So PDX models are generated when a fresh tumor tissue obtained directly from the patient and is implanted into the immune deficient mice via subcutaneously or orthotopically. So as such, these PDX models maintain the principal histological, clinical, and molecular characteristics of the original human patient tumor tissue while it remains biologically stable. Since PDX models are more close resemble and recapitulate tumors growth in human than standard in vitro cell line and standard other cell line xenografts, so they remain this PDX models become a key experimental platform for preclinical drugs development. So in our program, We will try to analyze breast cancer subtypes using PDX models. So apart from that, in this example, we will take a data set from a publication that is modeling precision treatment of breast cancer, which was featured in Genome Biology. So this publication is interesting one because the author assembled a data set of 50 different breast cancer cell lines that represent the whole spectrum of the breast cancer variation. So these cell lines have various omics layer data like genomics, transcriptomics, proteomics. So as well as the efficacy measurement to the commonly used in experimental drugs. So in our project, we will use a project of breast cancer subtypes, that is ER positive or estrogen receptor positive or triple negative. So what are these ER positive and triple negative breast cancer subtype? So ER is identified by the presence of estrogen receptor on its cell membrane, while triple negative has none of these three common receptors on its membrane. So this is just a one difference that makes one form of the cancer subtype is more aggressive, while other is as more treatable subtype. So how a biologist proceed for a research problem. So a biologist is hypothesis is largely mainly driven by the observation. So the observation give rise to the questions that can be definitely answered using data generated in an experiment. So this hypothesis can be confirmed or the refused based on the analysis that is outcome of an experiment. So the ultimately the collection of these results is exactly the what it's generating a knowledge. So what the big data changing the way we view results. So results are often at times the data itself that is reanalyzed by numerous groups and lab to generate a hypothesis before a new data is generated. So in comparison to the biologist approach, what could be a data driven approach? So now let's understand. So if a disease outbreak could be so let's have like if a disease outbreak could be forecasted like a weather, then a community could set up a protective measurement to control their impact. For example, at Los Alamos National Laboratory, scientists are improving disease forecasting mathematical model by using clinical data with various types of information. So their main goal is to develop a better understanding of how disease spread and eventually to able to predict a disease outbreak. So as we know, researchers are generating large volume of biological data, which has changed the way that biomedical research is done. An important aspect of a big data analysis is the ability to identify trends in omics data without any experiment or clinical mechanistic understanding. So typically, this is referred as a data-driven approach, where research is based on not just a hypothesis, it is based on the data that we have. So next, by using different machine learning tools, one can discover the relationship or the trends within data that are obviously not otherwise understood. So it enables the researcher to ask specific questions on the basis of the computer identified trends. So now let's understand how we can proceed for a data-driven research. So this process has 6 steps. It starts with identifying the question. So first of all, we try to ask a question, that is what are other alternatives to our perception. Next, we go for the review findings, that where we search who has already invested with this particular problem before us. So what approaches have been taken in the past to solve this particular problem. Then we design the model, or we formulate a hypothesis. How might specific variables can influence a the mathematical model that we are going to design or we are going to choose. So once we have these information, then we proceed to collect the data. that we have. So the most important, this is the most important aspect, specifically if you are working on a big data. So eventually we had it towards the data analysis, where we can apply various statistical methods to evaluate the correctness of the approach and repeat this procedure until the method has been identified. So at last, we frame the result, and that you can and finally frame into a story that could be provide a complete comprehensive research story regarding a particular research problem. So if you have any question till now, just post your question here. Do you have any question till now? Okay, so if you don't have, then let me proceed further. So, okay, I mean, by running the last session, pipeline in the last session, we have obtained this kind of table, right? So today we will be talk about the approaches that are important for the working with our data table of gene expression, and prepare it for any downstream analysis. So here inside each cell we find a quantified value that is called expression label. So depending on the input data, it could be depending on the input data or the quantification approach that we have selected, it could be a FPKM, RPKM or TPM values, or it could be a raw read counts. So today we will understand how we can analyze this gene expression table. So before we start looking for a specific gene, we need to summarize this data. So let's talk about some of normalization technique for the gene expression data. So first thing that we are going to not talk about is the box plots. So what do they actually represent these? What do this box plot is representing? So it shows different statistics about our data. It shows the median. Then it also representing giving the interquartile range with the lower quartile value as well as the upper quartile value. Then it gives us a minimum value and the maximum value regarding the sample, and it also provided information regarding the outliers, so that go beyond these values. So conclusively, with this box plot, we can get the summary statistics regarding our data. So we can do these kind of visualization of data using various packages in R. So can you tell me that you have access to the OmicsLogic platform? So let's go over there and see how we can visualize our data in the R. So do you have the access to OmicsLogic platform? Just go over there and let's log into the this particular server. Deepsa, can you provide the link for this particular server, OmicsLogic platform? Just share the link in the chat. So just let me know if you are on this server. Just post your answer yes or no. That's great, Madhuri. So, okay, so if you're able to log into this server, so if you click here on the courses, so just I'm giving you 1 minute so that everybody able to log into this server. So at this OmicsLogic platform has various tutorials by which you can learn how you can write a code in a R and the Python. So today I'm giving just a demonstration. Apart from that, you can also do it on your own if you have R Studio on your own system, or if you have Jupyter notebook on your system, so you can run a Python. So here we just giving you our platform where you can write your code and you understand how we can do all these complex steps just writing a simple R script. So here you will on this OmicsLogic platform have various tutorials that will help you to learn regarding the coding. So once we click on this courses, here you will find various tutorials. So let's start with introduction to the data science. So every one of you able to do this. If you able to log into this, able to just direct it to this particular lesson, that is introduction. Just go the tutorial introduction to the data science. So here you will have 6 lessons. So the lessons where there is no star that are freely available, while the lessons that are with the star that you have to subscribe. So as we want to visualize the data, so in form of a box plot or a heat map, so a good image with the simple image that is that will explain lot of regarding a data. So you will learn about basic visualization package that includes box plot, scatter plot, histogram, wall plot. So now let's go over to this data visualization lesson. So under this, in every single lesson, so you will find different steps. So are you able to access to this particular lesson? That is data visualization under introduction to the data science. Okay, that's great. So here with step-by-step explanation is given how you can proceed a lesson. So let's start it with the first step, that is showing the understand the problem. So the first step will explain you what we are going to do in this particular lesson or the tutorial. So like here we are going to visualize the data in form of a box plot, or it could be a bar plot, it could be a heat map, it could be a scatter plot. So then uh, here the in a step by step explanation is given. So how but is each step is doing. For instance, under the first, so let's data cleaning in R. First, let's load a data and view it. So first step, with the first step, we are actually like with this command, what we are going to do, like data equal to read dot table, then you have to provide the link to a particular file, right? So then we have to this is the Excel file. So then we have to tell whether the information is like in whether there is header or not in the file. So apart from that, you will also provide if you have tab separated file or the comma separated file, so then you will give that information with the separated. So this lesson will teach you that what we are doing with each step. So once you understand about these steps, then you can complete it, and then we can proceed to the next step. So under the next step, that is about learn the code. So here code breakdown is showing in different steps that how we are actually going to perform our analysis, or we are going to write a code. So once you this is how it will show like here. So first you are uploading your data, so then you can check whether your data get uploaded or not. So then with this box plot and the heat map, you are producing these box plot and heat map. So then with the bar plot, you can visualize the data in form of a bar. So let's complete in and let's go to the next step. So now here, let me just do. So first what it is doing, here we are uploading a data. So here we are already providing a path. Here you can even provide your path of your own file from where you want to analyze data. So what it's doing, we are storing our data. So with the command read.table, you can read your data. Then here you will provide the path of this particular. Let me just copy this. So first of all, you need to provide the path of the file that you want to upload or import. So then we can tell whether it's a tab separated file or it is a comma separated. And then we are then we are telling the whether there is header or not. If true, if a header is provided in your file, just write a true, otherwise false. So now let's upload this data. So how we know that this step is already run? So it's showing that success running the code. So now how I can check whether my data is get uploaded or not? The one way is I can just print this or view this, view data df. So one way is I can view this data. So it's unable to view. So the other point is just do it df. So it will print your whole data. So like here with the df, we able to write this data that is in the console. So we able to check whether our data is get uploaded or not. This side you can even with the head command, you can check whether your data get uploaded or not. So this is how you can check whether your data get uploaded or not. So then just next go to the next step where let's create a box plot. So as we have already uploaded data, now I'm just skipping all these steps. So first of all, as we just next we want to visualize this data in form of a box plot. So with this box plot, so you can visualize your data. So it's indicating that there are the normal samples. So then this is how it looks like. So with the box plot, then it is the apart from that, then we can use a heat map. So these heat map represent, now we can visualize our data in case of the heat map. So here what it representing, like how these genes are expressed in different samples. So like here samples are given. So here the different 15 genes that are given. So with this actually we able to visualize. Anirudh have a question. So tab separated. Okay, so some of the files. Okay, let me just show one thing. Just give me a second. I will just show you what is meaning of tab separated. Just let me just complete this lesson so that then I will let you know. Like some of the files are comma separated, so these are the separators. So it could be a tab separated file, it could be a comma separated file, it could be an Excel file. So I will show you that each column is actually separated by a tab, not with the comma. So that is what a tab separated. I can show you, Anirudh, what kind of files that we could have, and but a tab separated file. So what this heat map representing, so it summarized your data like here in different samples, but we can also even gave the name of these genes. So that's a different options, and we can explore those kind of options ourselves. So then it's like it for this particular first gene. So how this particular gene is expressed in different kind of samples. So sample is given in x-axis, while these genes are given on a y-axis. So you can visualize the expression pattern of these genes in a different samples using just a heat map. So this was the box plot. So then we can also visualize our this gene expression table in form of a bar plot with the command bar plot, then df. So if we run this code, then you will find various kinds of bar plot. So this is a very simple way how you can write your code. Just let me so in form of a bar plot we can look at this. So how these genes are getting expressed in different kind of samples. So this is how we can visualize our samples or our data using various visualization tools, like it could be bar plot, it could be a box plot, it could be a heat map. So here what we have done, let me just do it. First, we have just given the file, like we just with the read dot table, we read this input file, and then we just remove the first header. So then we converted, as these box plot or heat map are actually taking not as a data frame. So it is reading a data in form of a matrix. So in this command, so let me just remove it, df equal to as dot matrix. With this command, we are actually converting our input data in form of a matrix. So then with the box plot command, we are just uh, finally we are generating a box plot of our data that is stored in a df. With the heat map, we are storing our data in form of a heat map. So then the bar plot command, we are just storing our data in form of a bar plot. So this is how you can visualize your data, and you can understand what kind of the patterns you can see in this sample. So for instance, at this particular heat map, so you can see this particular gene is actually comparatively differentially expressed in comparison to these other genes. So this is how you can visualize your data and you understand about your data. So then we can complete it and move to the next. So once you complete it, you will also get some assignment. Here you just need to give feedback regarding this chapter lesson. So like here it's asking my name. So once you complete it, it will complete your lesson and you will gain different various points. I pick the first name, then I have to give. Then you have to provide your email ID. So then it's asking whether it's difficult extremely, or I'm just saying extremely easy. So once you're able to complete this lesson, so then you will get various points for this lesson. So beside one more thing, I just want to tell you, you can run these codes on your own Art Studio or the Art platform or the Python platform. So there are the different scripts that you can download. Just give me a second. Okay, so here you will find an option like this one. So this will it will copy your code directly. You can copy this code and then you can paste on your own platform. It could be R Studio, it could be R platform in general. So you can copy this code directly from here. So then you can run this code on your own platform. So here you just need to provide the path of your file. So if you are working in that particular folder or working directly, so in that case you just need to provide the name of the file. So this is how you can learn a simple code in the code platform. So every one of you able to complete this, just let me know. So one more thing. So suppose here we are just want to, so you don't want to run every single step. So with the hash, you can comment it. So like, let me just do it. Suppose here only I just want to draw the box plot. So I can comment other things, right? So here you will get something is wrong with this code. Apart from that, I think missed something. So every one of you able to complete this assignment. So you can comment other that suppose I just don't want to generate a heat map. I don't want to get a summary statistics. So we can get a summary statistics as well. So just comment it others. I don't want to generate a box plot. I just want to get a summary of this particular DF. So there is some problem. Okay. Let me just go over this. I just done something wrong here, I guess. Okay, so here, even here, we can just find a summary. With the summary, we will get the details, summary statistics of this project. So yes, it will provide us what is the mean, what is the minimum value, the first quartile value, then what is the maximum value. So all those statistics you can get with this summary command only. So then you can draw a box plot. So just let me know whether any one of you able to complete this, able to run this by this code. Just post your answer in the chat box. So this is, if you're not able to do this, just go to this lesson and try to complete it before the next session. So now let me go over the presentation. So just let me know whether any one of you able to visualize your data that is available in the platform. So I'm looking for your answers. If you can, if not, yes or no, just apply, at least just reply that. Are you facing any issue in accessing or something? So just let us know so that we can understand, are you facing any problem? Besides, if you're still facing in your problem and you're not able to do something, just post on the forum. So you can post your query at the forum of this organization or this program. So now let's move ahead. Okay, so when looking at a gene expression table, a large number of samples will look like the outliers. At here, we can look at here. So this type of distribution will interfere with the many of the statistical tests that are applied to analyze any gene expression data. So to reduce this wide range of variation, we can transform our data table or gene expression table into the log normalized values. So now this data become a more normally distributed, and this procedure of normalization is called as log scale transformation, and it makes the data normally distributed and more symmetrical, for where we can apply various statistical analysis. So how it looks like the initial data. So like here, when the data is not normalized, Then once you normalize your data, so how it looks like here in the figure 2. So now let's look how this log scale transformation affect the gene expression pattern in our data. Let's take a example of one sample. So in the raw data, we have lot of genes that are low expressed, while there are only few of the genes that looks like highly expressed. So after the log scale transformation, this data become a normally distributed. So if you belong to you are statistics student, so you will know that a normal distribution means it follows a bell-shaped curve. So after the log scale transformation, this data become a normally distributed. So now it's easier to compare the values between the sample without losing the details of low expressed genes that here looks like, and still see the highest and the lowest values clearly. So it will be very useful to make an accurate assumption regarding our data. And now we can apply various parametric statistics tests on this particular data. So another useful transformation or the normalization method that you might consider specifically for the NGS RNA-seq data analysis. So it is the quantile normalization method. So this is most useful when you have number of the conditions and you would want to compare, assuming the samples in each of these conditions are actually biological replicates. So as in this example, we can see there are multiple samples in each of these two conditions. These two conditions, like here, one condition is looks like a light green color. The other condition is looks like in a light orange color. So what this quantile normalization is doing, so it transforming or normalizing this data and arranging the expression values in similar order between the samples and align their means. So this type of normalization help us to understand the different kind of the genes within our data set. So how it is doing it? So now if we looking at this particular figure or this particular plot, so the majority of the genes are under the have normal gene expression despite the condition, whether it's a condition 1 or condition 2. So the reason is that these genes perform a normal function within the cell. Then we have few genes that have low expression. We can take consider them as technical noise. Then we have some set of genes that have a significant signal. So and these statistically signal, so the values are represented here. So then these genes are important ones for which we are looking for how they actually differentiating two different disease conditions. So what would be the effect of the normalization? Let's take one more example with the quantile normalization. So here we have we have the samples from two different categories, that is ER positive, that is estrogen receptor. Then we have a triple negative breast cancer subtype. So if we looking at this raw data, it looks like this particular gene is highly expressed in one condition, that is ER, and while it is low expressed in the triple negative condition. So this is before the normalization. So once on the normalization, this difference just disappears. So as a result, some of genes that do not have very strong differences in the expression will be less significantly different. But the overall difference between the samples will not change. So this is important for the noisy data, where thousands of the observation about each condition can be influenced by various technical variables or technical errors. So this shows why normalization of data is important to drive a correct biological interpretation from the data. So now let's see how we can perform log normalization of our data in Excel. So let me share one link with you. We are just performing the log normalization on the data that we have obtained yesterday. So just let me share the link. So just download this data. So this is a gene expression table that is obtained from the raw sequencing. Eventually, we have our data in the form of FPKM values in form of a gene expression table. So if you copy this data, so just let me paste this data and let's see how we can perform log normalization just in Excel. So why we need to perform log normalization? Just look at this table. So this table, if we just looking at this table, so just take a look here, give a look here. So like here we have the values like 0.054, and then we have a value like this is 13,707. So like 15,000 values. So this is like a very large variation within even a data set that so that's why it is important them to transform into the log values, so that we can bring our data into a particular within a small range. So every one of you able to download this data. So just paste that data in Excel. And just once you're done with that, just let me know, just write your answer in the chat box, yes or no. So any one of you able to download this data and able to paste it in Excel? Whether you are not able to download this data or but the issue that you are facing. So anyone able to download this data, just let me know so that I can proceed. Okay, so are you able, great Madhuri. Anyone else? Anirudh, that's great. Okay, so if you able to download the data, just paste in an Excel. So now let's look how we can transform this data into the log normalization. So another factor that is very important, let me just tell you that. So as there are, if we looking at this data, it contains zero values. So if we take a log normalization of a zero value, so it won't give it don't give any numerical value. So for that we before just performing log normalization, we need to add uh, one value, because if you take a log of 1, it will become automatically 0. So let's just go here and let's see how we can perform log normalization. So there is a simple formula, just write equal, then just do ln. So then you have to provide a cell that is v2. So then v2 is the value that we are just giving. So I'm just providing here uh, the cell which contain the particular value, gene expression value, and then if I add 1, so then I'm able to do log normalization. So now let's look as v1 which column. So here I just v2 So this is V and 2 is this. So I am taking log normalization of this particular value. So now I can expand this log normalization as this is taking a V. So the last column that we have, that is U. So U2 will be able to, so we just drag this value. So let's see. So S, so then again we can drag S. So till the we have these values. And now let's, as it converted the first row, so now we can take them together. Then we can drag this, and we can all drag this till the end. And one method is you just click on this end. So this is how you can completely log normalize your data till this value. So now if you're looking at these values after the log normalization, the values that we are looking here like a 13,000, 15,000, right? So now if you're looking at those values, so the now range coming, these values coming within a range like 9.5, 9.6, 9.6. So and another, then we have the value that we have 1 point something, it is 0.3. So this is how we can actually transform our data that have very wide range of variance into a simple log normalization. So every one of you are able to get this point. So are you able to perform log normalization in Excel? So you can perform this log normalization into R as well, just a simple function that is the log. So in that case, you have to add, like we have data in the DF. So if you do that, DF plus 1, then in the bracket, then you just write a log. So in that case, in R, it is a very simple, and even in the Excel, it is very simple. You just have to write equal to ln. Then you need to provide the cell that you wanted to convert. The cell that you wanted to convert, like here we want to do V1, then just add a +1. We here adding only +1 because we have lot of zero values, and if you take a log of the zero, so then it will doesn't give you any output. So this is how we just can perform a log normalization. So any one of you able to perform log normalization in Excel, just let me know. So just write yes or no in the chat box so that I can understand, do you able to perform log normalization in Excel? So this is how it's pretty, that's great. Anyone else able to perform log normalization of her data in Excel? Apart from Preeti, anyone else able to perform log normalization? So it's better if you just reply yes or no, so that we can understand you are getting anything out of it. Madhuri. Okay, so what's the problem that you are facing? Just let me just repeat it. So what you have to do, you just paste your data in Excel. Then, okay, so not, let me just do it again. Not open to an Excel. So one thing you can do is just, so let's do it again from the beginning. So We have this data, just copy it, Control A, Control C. Once you are able to do this, just open your Excel and paste it into a new sheet, Excel sheet, right? So if you're not able to open this file, as this is a text format file, so might be you facing some problem. So you can paste it directly, just copy and paste it in Excel. Madhuri, are you able to do this? So once you paste it, so I just check it from where our table started. It is not formatted. Okay, so you must be using a Mac. So in that case, so okay, and you just need to perform log normalization. You can take even just a some rows or some seats. So I think it is, if it is enough for if you're using a Mac, then you just need to paste it in, like you have to select that tab separated, delimited. So the one way is to do that. Here. So like here you will get an option, right? So this this option, if you click on this, you can just use text import. So then this text import will asking you the delimited. So then proceed for the next. Then here, as our delimiter is a here in this case is tab. So with this tab, by selecting this tab, you just proceed. As you know, here you can see that you are able to your every single column and row is getting separated. So you just finish here. So this is how you can paste a file. So here, like once you paste, you will find this option here, this option. And if you click on this drop down, then you find here use match destination formatting and use text import. You can use text import wizards. So once you select that, you will get this window. And this window first it is asking delimited, just proceed to the next. Then once you proceed to the next, so as this file is a tab separated, so you just finish, you can finish this here. Once you're able to do this, so your table will be get pasted here. So okay, you can take just a few example. I just trying to, if you're not able to do this, we can address that problem even later as well. Madhuri, now you're able to just, Madhuri and Renuka. So now you're able to paste it or not? Okay. Okay, so then you just have to write command ln. So then we have to look in from where our table is getting started. So if my table is starting from this B column and 2 second row. So I am selecting with the cell B2. Why I have used here B2? So we can then with the ln, then within the bracket, we can define that cell plus 1. We are using here only as we have a 0 value. So we are adding 1 into every single value so that we can get, we should get some numerical value specifically for the 0. So this is how you will get. So if I don't add 0, what it happens? Let me just try, let me show you what it will give you like this. ln. So here I am just selecting this particular column, and if I take the log of it, so it won't give any numerical value. So it will show me this error. So to resolve this error, that's why I have added ln. Can select this column plus 1, and log of 1 is 0 plus 1 become 1, and log of 1 is 0. So any one of you able to just able to just log transform this table, are able to, so then we can drag till the columns that we have. So we can drag towards the column as well as so we have last column that we have used. So till that we can just drag it towards the column, and you can also the last column that we have used. So then after selecting, so we can also drag it to the in the rows depending on our values. So this is how we can perform log normalization in Excel. So I think we have we are already over time. So let me just proceed further. Just let me close this session, and you can post your questions even on the forum. So then I'm just giving you some few assignments for the next session that what we have done today. So you just go for that first. If some of you are yet not completed your profile, as already Beepsa has shown you, so please complete your profile, write your name, upload your picture, and you can also link your this profile ID with LinkedIn or other social media sites. Then complete your introduction to the bioinformatics course, and as well that the part that the end part that we have done, that is the part of the transcriptomics 2 course. So just start your series, this transcriptomics series. So as we are going to perform all those analysis on various kind of analysis, and those are very well explained in the transcriptomics 1 transcriptomics series. So the session that we have covered yesterday is covered under the transcriptomics 1. Today we just begins with the transcriptomics 2. So just start these courses, to start completing these courses. And once you complete your course, at end of, always finish or the complete your course button, always click on that. Otherwise, even if you have completed your course, it will it won't show you that you have completed your course. Then just explore the coding lessons on this Code OmicsLogic platform, particularly just how we can visualize or load our data on R. So just complete it before the next sessions. I think you will have 2 or 3 days. So just try to complete these lessons and try to run the pipelines that we have run yesterday on T-Bioinfo platform. So if you have any questions now, you can ask, post your questions in the chat. And in the next session, we will discuss how each type of data is associated with the specific steps designed to eliminate technical noise and links direct to the raw outputs from an instrument with a studied element contained in the knowledge databases. So in next session, we are going to understand the cleaning, loading, and the processing of the data. So if you have any question, you can post your question in the till now from this session. So now we are ready to address your questions.
Thank you so much, Dr. Harpreet, for today's session. So, as Dr. Harpreet also mentioned, please go through Introduction to Bioinformatics for most of you who have not completed the Introduction to Bioinformatics lesson, and then start with T1. It will take you some time to go through these lessons together, and our next session will be on Friday. So if you do not have any more questions, then for today, we will be ending our session right now. And please keep using the forum and posting all of the different questions that you may have on the forum. I will again be sending you the video of this session as well as all the assignments that you need to complete, and share that email with you as well as post it on the forum. So reach out to us with any questions that you have and start using the forum as well. So by the next class, everybody should have their profiles updated and at least the introduction to bioinformatics course should be completed. I think most of us can do that, right? It would not take you a long time. And for anybody else who is facing a problem, if you're having, like, if you do not have anything like an access to any of the platforms, please email me and let me know that you do not have this access so that I can look into it and we are ready. Because as we go forward, we will be going through more advanced lessons. So this week we can actually help sort out any kinds of issues that you will be having. Dr. Harpreet, your screen is still shared. Yeah. So thank you so much, everyone, for joining today. We will meet you again in the next class.